110. So efficacy will be 0 if the quality is 0. And now these days we have been facing so much of challenges in getting the raw materials. As Dr. Honeyman directed, what Dr. Honeyman said was a physician must be provided with genuine drugs. Genuine drugs remedies can be provided only when we get uh, genuine raw materials. And this is a very big burning problem in the field of herb, especially I would say that in the preparation of vegetable kingdoms, getting the right herb is a very big challenge. Many top level international journals have highlighted, you can see in the screen, that they are stating that adulteration and substitution of crude drug is a very big problem, burning problem. And not one single journal, there are so many journals which have highlighted this point. So what we do in the homeopathic quality control, and especially which at Schwabe India, to ensure that we procure the right raw material and we are preparing the subsequent products, the dilutions, higher dilutions from the right raw material. So if we take the herbs, there are so many adulteration. I mean, so many substitutes we are getting in the market. If we ask for example, for some herbs, we have our own cultivation. I mean, Germany, even in India, we have cultivation plants. And in Germany, it's one of the most diverse cultivation plants in the Europe. And we grow almost every homeopathic herb which is required for production. And in India, it is the country itself is a very diverse type of country. If you go down to south, you get many plants which can be cultivated in high degree of temperature. And if you go to Kashmir or some other places of Himalayas, then you can cultivate actually the herbs which can be grown in a very cold climatic conditions in Europe or North America, such kind of things. So in India, the problem which we have been facing is we don't get always a good quality or the right material, right species. For example, I have given some examples here. If we ask for hydrastis canadensis, then Thalictrum foliosum has been given to us. And we check it and we find it is not hydrastis canadensis root, so we just reject it. And in the same way, it is true with Senega, Genosia Ashoka, Sabina, etc., etc. And how we ensure that this is the same raw material or not. Yes, I'm before coming to that. You can see here there are two different raw materials. One is Equisetum hymen, another one is Equisetum arvens. Both would look almost similar, but if you do some tests, then only you could be able to identify and find that this is not the same raw material. They are different because both are from the same genus, but the species is different. Our Materia medica has even a very minute indication which is pertaining to particular species. So you cannot use Equisetum arvens in place of Equisetum hymen. And basically, if we see the procedure, what we follow is we receive the raw material from from outside. I mean, from whether from our in-house own our cultivation plant. In case of our own cultivation plant, we are 100% sure that in the beginning itself, the seed itself undergoes a thorough quality control check, and we make sure that only we get the right species of the raw material of the plant is grown. But when we get from outside, I mean, there are certified third-party vendors who procure raw materials from other suppliers, other cultivators, and supply to us. And in such a case, when we receive the raw material, after receiving it, it is verified by the quality department and also the raw material storage department combinedly. And then that has been sampled, sampling has been done, and the quality department thoroughly check its sample whether it is correct or not, and then they go for they whether after completing the QC testing they would either approve it or reject it. So that is how the process is followed. So what are the parameters they generally use to check that particular drug material, raw material, whether that is genuine or not? there are many parameters, but they are different and individual specific to each and every herb in many cases. And for example, at the beginning, they do macroscopic examination. I mean, just with naked eye, they will check whether it is a right plant or species just at the first sight, and then whether the the part used of that particular plant is correct or not. Many times, this is another problem we get, that we get the raw material, same raw material, but mixed with some other parts. For example, Abroma radix. Abroma radix is the root of Abroma tree. Abroma, it's a kind of a shrub. And when we get the root, it is not completely root. Almost 30-40% the harvesters harvest it above the root. So when they cut it, when they cut the raw material, they don't take it only the part of root, and they just cut it 1 foot higher than the field. Then they just supply that directly. They have to actually remove the upper part and send us only the root part. But many times it happens. Then what we do, we either send it back to them for rectification, or we do on our own. I mean, we use our own manpower. We get it separated. We cut it and separate only the root part, and we charge the people, the supplier who had supplied it for those manpowers. And this is how it happens. And in this case, yes, we can accept, and we can. Our quality department will approve that particular raw material with the condition that the upper part of the stem part has to be removed. But in other cases, it may not be possible. We get the right species, exactly right plant, but it is not in a good condition. Many times, what happens, there will be some. It is stored in a very humid temperature, and it is not dried properly. So in such case, there would be a fungal development. So we generally what do, we would reject that, and we will ask the supplier to take it back, or we would destroy it at our own pace. So in such condition, there is no conditional approval. It is completely rejected, outrightly rejected. And then they check the purity, whether is there any other foreign matter. Many times what happens, there will be a mixture of soil, sand. So such things can be approved with the condition that that has to be cleaned. And this is how in the beginning it is done. I mean, with the macroscopic examination, and then it goes to microscopic examination in case of fresh materials. They take the cross section and they check whether with their database whether it is matching with the same species or not, and then they go for HPTLC fingerprinting. It is high performance thin layer chromatography. It is a kind of fingerprint for the plants. You know, if you have your own fingerprint and there is your friend's fingerprint, both will never match. In the same way, the fingerprint of TLC fingerprint, HPTLC fingerprint of each herb would be different. For Abroma radix, it would be different. Then for Abroma augusta, it would be different. And then apart from that, we do UV also, UV spectroscopy, and again the UV absorption would be different for each plant material. So that is another way how we ensure that it is same or not. For example, if you take the fingerprint, this is, for example, there are 4 samples of one particular plant, 5 samples, 4 samples here. It is there, it is shown, and you can see at the bottom, all are looking almost similar. Here at the bottom, it looks almost similar. But if you see the color is different. So it cannot be the same. But if you see these bands, you can clearly see that all are different. All 4 are different, because here you can see there is a similarity of these bands, but the color is different. And here also there are some similarities, but the colors are different. So you cannot say that both are same. Same. It is completely obviously it is different. And in case of the same herb, what happens? This would look exactly same. For example, this is our control sample. This is for just to compare with the test samples, and I have 5 different samples, and I'm just checking this with the control sample, and I could be able to see that these test samples are matching with this control sample. So it is very clear. So it can be accepted. So finally, this sample is accepted, approved by the quality department, but the other samples are rejected. And we many times we say there is a saying that seeing is believing, but it is not always true. Because here you can see there are I have shown another picture before that both plants look same. Here also in this case these are some of the raw materials which have been rejected by our drug control department, our quality control department. And for example, I would highlight one particular thing which is here. This is, you can see there is a root plant material here. Uh, Root material here. This is actually, there are two samples you can see clearly, but these two samples are looking same but different. This is actually Vidania somnifera, ashwagandha root. But what happens here in this case, it is same ashwagandha, and ashwagandha again from Vidania genus. The species is somnifera, Vidania somnifera. That is also we have checked, it is same. And we checked, is there any fungal growth or not? We found that there is no fungal growth. Condition is good. It is looking good and also healthy. And then we checked the fingerprint. We took the extraction from this particular raw material, both the raw materials, and we found it is also good. But finally, we found that one sample is that is our own sample. That is a that is a sample for checking for the comparison, and the other sample, test sample, which we have received from the market, was actually already extracted. It was a pre-extracted sample which was sent to us. I mean, what they do, they just put it, soak it in different solvents, ethanol, ethyl acetate, or whatever may be, and they just take out the vidaniferin, vithanolides, which is their which is the active constituent of this particular plant, Vidania somnifera, and they have just removed that and they have supplied. It is looking like same, everything is good, but the extract has been taken out. So if we prepare a homeopathic mother tincture with this, then definitely the quality would not come as desired. So you can see how the herbal industry, as the technology increases, people who are supplying, people who want to actually cheat, they are also improving their technologies. So this is another thing. And if you take mineral kingdom, the same way there are different types of methods we follow to ensure that the plant, the particular sample of that mineral kingdom is right or not. Starting from the description, whether the description is matching with the sample, then identification, there are identification tests, then solubility, its odor, then limit tests, whether the percentage is right or not. Then LOD, the LOD is loss on drying, how much moisture is there, that has been checked. Ash content is checked. A particular chemical assay for that particular chemical mineral drug, that has been checked. Melting point is also checked in some cases. Specific optical rotation and refractive index are also done. And yes, in some cases, UV analysis, ultraviolet analysis has also been done. And in many of the cases, we also check microbiology test, because to ensure that is there any contamination with this sample or not. It will make sure that no, it is pure raw material and it doesn't have any microbial contamination. And if we move ahead with SAR codes, in case of SAR code also we need a certain, we have to do check and ensure that the raw material is correct. And it has to be matching with the description which is given in the pharmacopoeia or in-house developed standard. Then identification starts from the beginning itself, when it is extracted from the plant, from that particular animal itself. I mean, in such a case, what happens, that a veterinary physician would make sure that this is taken from that particular animal species, and this part is the same which is described, which is given in the description. So that is that. And then apart from that, if there is some mineral in that particular gland or sarcode source, that also has been identified. That has also been not only identified, also measured. How much quantity is there? Is there a desired quantity? For example, in case of thyroidinol, we check the quantity of inorganic iodide. How much is the iodine content in that? And then apart from that, acid insoluble ash, fat content has been checked, because fat content varies from particular animal to animal, and also particular gland to gland or tissue to tissue. And then a particular assay which is required for that particular raw material, particular glandular substance or whatever the sarcode substance, that has been done. And then microbial contamination has been tested to check, to ensure that there is no contamination, because in these cases, animal products, there is high chance of microbial contamination. Then LOD, laws on drying. I mean, it is basically the estimation of moisture content, that also has been done with sarcodes. And if we look out no source and imponderabilia, it is very difficult to make sure the raw material is correct or not, but we can ensure the process. I mean, if the process of preparation is in front of you, then you can make sure that it can be done. For example, in case of no sode, yes, we have in case of no sode, when there is a microbiological substance, yes, it is possible. Raw material, I mean, the raw material of the microbe has been tested and confirmed, that is possible. But the no sode stock solution should be prepared by your own. I mean, we have to prepare on our own. You cannot make sure after preparation of the stock solution, it is not possible to again ensure or check or reject that particular stock solution. Yeah, because already you have diluted that multiple times. And it is that making sure of the quality control or standardization of raw material is limited only to the quality assurance during the process of the preparation of the first initial potencies, initial stock solution. You can make sure that the process is followed properly. And the same is true with the imponderabilia also. Imponderabilia, for example, if it is X-ray, you can make sure that the X-ray machine is with this particular specification. That is possible. And also the X-ray wavelength is this much. That is possible. But once it is prepared, when you prepare the stock solution, that is not possible to ensure that it is prepared from the same that particular type of machine or not. That is not possible. So it is limited only with the quality assurance. And we need to pay special attention for some specific special raw materials. For example, calcarea carbonica, many people think that you just take pure calcium carbonate and prepare, that would be carbonica. No, that is wrong. Actually, it has to be prepared from the oyster shell, because oyster shell has many other ingredients apart from just calcium carbonate. So it has to be prepared from the oyster shell, and then only when you apply that particular remedy to a patient who has the characteristic of calcarea carbonica, then it will work. Otherwise, if you prepare a dilution 30 200 1 m from the pure calcium carbonate, then it may not be working. And then in case of heparsult, we have to prepare oyster shell mixed with sulfur in a specified way, as suggested, directed by Dr. Handyman. And in case of phytolacca berry, uh, fresh berries have to be procured. You cannot, in case of many imported drugs, it is allowed to import the dry raw material, dry part. But in case of phytolacca berry, it is best to procure and transport fresh material. Actually, it is growing. It is grown in North America. We don't have in India. And when you transport it, it is actually impossible to transport the dry raw material. But we have some arrangements with some transporters so that it will be prepared, it will be procured fresh, and then it is preserved in the menstruum, and it is transported. It is costly process, because when you import dry raw material, then the cost would be lesser. But when you are preserving it as in menstruum and again transporting, then the quantity increases much, and accordingly you have to pay more to the transporters. And Apis mellifica, in case of Apis mellifica, there is a special method to prepare that. For example, you cannot just take honeybee and just prepare the do the percolation or maceration and prepare the mother tincture. It's not possible. You have to procure the honeybee, live honeybee, and that bees have to be put in a cylindrical vessel, stainless steel, and you have to put the menstruum at the bottom. Then you have to shake it vigorously. During that vigorous shaking, these honeybees would go and spit out the venom from its mouth. So that is actually the active ingredient, which is actually helping, acting has a therapeutic efficacy. And Calcarea ovatosta, it is not just the whole egg, it is just eggshell. You have to take out the eggshell. And in case of arum muriaticum and arum muriaticum naturatum, again there is a special preparation of gold chloride, and it has to be prepared from that. And for the pharmaceutical industry, water and alcohol is also a raw material. You cannot say that they are just vehicle. In our pharmacy book, you might be studying it as vehicle, but for the pharmaceutical industry, it is actually raw material. So that also has to be tested. What we do here is we check the alcohol quality twice. I mean, first what we do. I mean, if you see here, uh, there is a before procurement of alcohol, a test report has been sent by the distillation unit. They will send that, see, this is this particular batch alcohol's test report. You can just see and check, and we check it. And then again, what happens, we again go there before procuring, and we get the test done on site, and then we bring the alcohol. Then again, when we are going to procure the vehicle alcohol, so actually as per our SOP, we bring the vehicle tanker, alcohol tanker, to our plant first. Then it is thoroughly washed with the demineralized water. Then our Shobe India person will go along with the tanker and get it filled, and he will bring it back. Because what happened, we had past experience that we always we get the tanker here and we clean it with our demineralized water, because we are not sure which water they use, other people, outside people, so that it will be contaminated. uh, but what happens? What happens? What happened once? Actually, we had some experience also. We got the tanker and it was cleaned in our plant, and then it was sent to the distillation unit, and it again came back. We took the sample of alcohol and we checked. It was contaminated. Then we did a root cause analysis, and we found that after we cleaned the tanker, that guy in between, he loaded some other chemical in his tanker, and he went to the before going to the distillation unit, he unloaded somewhere near distillation unit just to make money. That I mean, if it goes empty, then he will not get anything, even though we are paying them double. But he wanted to make extra money, so he unloaded and he didn't clean it properly. He didn't wash it properly. So when it came back to us, then the entire container was contaminated, and we had to reject completely. Then from there onwards, we started this particular process that our person will be accompanying the tanker after washing at our plant. So this is how the process is. And yes, water also is an important raw material for us, because in case of dilution, only alcohol and water are the raw materials. Other our homeopathic remedy is basically, it is a, these vehicles carry the dynamic therapeutic properties of the drug, and we take the raw material, raw water, and then we bring the impurities from almost 2000 parts per million ppm of TDS to 50 ppm. That is RO water, reverse osmosis. Then from that we prepare demineralized water, which is less than 10 ppm. Then we prepare distilled water. It is just H2O, nothing else. And here also we do daily fresh water. We use fresh water on daily basis, and that water is kept on circulated. Then apart from our own internal test, we periodically send our water to external labs to make sure that it is our testing process also is working or not. So this is much more important. Then apart from that, we have to follow certain additional standards in some cases. For example, in case of triturations, we measure the particle size at 1x level, 1x trituration level after completion of the trituration. And if the particle has not been reduced to less than 10 micron, and it has to be again, it will be triturated for some more time. And then apart from that, there are many other things which help to maintain good quality product, not only after passing the raw material, also the complete system, quality system. It is called as TQM, total quality management. And here in the entire factory, we check the microbial growth on regular basis, periodic basis, and then we maintain clean environment. Then workers cleanliness has been maintained, and the canteen and food quality has also been thoroughly checked by different teams. Then you have to, apart from GMP, good manufacturing practices, other good practices, GXP, anything, any good practices, you can put under GXP. So we have good laboratory practices, good warehouse practices, etc., etc. And we have a proper technology transfer. Whenever we develop a product, there should be, you cannot do like you develop a product with certain raw material, and you have not transferred that particular uh, product product development technology to the production unit. So then your product would fail. So properly the R&D department will transfer the technology to scale up in the production department. Then the quality processes are validated. We do self inspection, and vendor control is very important. And you have to actually have a process driven system, not a person driven system. And actually quality has to be integrated in all the processes. That is most important. And here you can see you have to have a proper ERP system to ensure the data quality. Then process should be controlled at each step. Electronic record and electronic signature would significantly reduce the errors. And also validation and audit trail has to be followed. And there is a, particular process which is defined in the US FDA, that is Code of Federal Regulation number 21, that is called as 21 CFR. And we have a 21 CFR compliant equipments. 21 CFR means that the chances of error is very less. That is what the objective of that 21 CFR. And then advanced quality equipments are required to ensure your quality of the raw material. They include GCHS with 4 decimal accuracy. I mean, it is not just 5.4 is the accuracy. It is just 0.0001, that much, 4 decimal. And HPTLC densitometer, again it is 5 decimal accuracy, 0.00001, that much accuracy. Then you have to have highly skilled manpower, and fortunately we have people trained in Germany. And analytical methods should be connected with the LIMS, it is Laboratory Information Management System, which gives us seamless quality management. And the microbial lab has to be at par with the current technology, and also the stability also has to be with the current technology with international access control. So these are some of the important aspects of the standardization of raw material. I thank you everyone for your patient listening. Thank you so much. Over to you.
Uh, Thank you so much, sir, for an amazing session. I would like to take a moment to express our profound admiration and appreciation